Hey apes, today we're going to talk about human healths and environmental risks. That includes risk assessment and toxicology. So this is actually a figure from Friedman and Relaya from your textbook, and it breaks out the leading causes of death globally. If we take a look at how this all breaks down here, you'll see that, of course, the number one cause of death worldwide is cardiovascular disease. But infectious disease runs a close second at 26%. And this is based on 2004 WHO, World Health Organization numbers. So things are a little bit different today than they were, although I couldn't really find a good pie graph that showed that. I could find some WHO in the data in bar graphs, but it wasn't quite as impactful. So we're going to just go with this. If you break out those infectious diseases, you can see that we've got respiratory infections at 30%, HIV at 21%, diarrheal diseases, tuberculosis, malaria. So those six are the six biggies, six biggest infectious killers, and we're going to actually talk about those in turn. Coming back over to the large pie graph, we've got cancers over here, also at 13%. And you know that cancer risk is multifactorial, meaning that there's many different causes for cancer. DNA damage underlies the development of tumors, most tumors, 99% of tumors, but the cause for DNA damage varies. By the way, this number, when I looked at the 2017 data, injuries, particularly traffic and road-related injuries, have gone way up. They're a larger percentage, and I didn't read the whole WHO report, but there's probably some reason for that. So just some terminology to set us up. A disease is any impaired function of the body with a characteristic set of symptoms. Infectious diseases are diseases caused by pathogens, bacteria, viruses, protists. Generally, those are the three biggies. It can also include prions, and we'll talk about prions later. Examples include pneumonia and sexually transmitted diseases, anything that you can catch from somebody else. Non-infectious diseases are not caused by pathogens. Examples include cardiovascular disease, which can be genetic, can be lifestyle, or a combination of the two, respiratory disease, digestive disease, and cancer. Acute diseases rapidly impair the functioning of a person's body and may cause rapid death. May cause, may not. I mean, if you have acute bronchitis, that means it's currently in an active state and it's rapidly impairing your health, but you're probably, here, at least in the United States, you're probably not going to die of it. Chronic diseases slowly impair the functioning of a person's body. These are diseases that that either take a long time to develop fully, or are exacerbated in conjunction with lifestyle, like heart disease. Cancer would be chronic disease as well. Many factors cause people to be at risk for chronic diseases. So these are cancer, diabetes, cardiovascular disease, and chronic infectious disease are considered chronic. And again, when you look at these individually, there's many risk factors, and the risk factors differ between low-income and high-income countries. So let's look at why and how. Low-income countries' risk factors are generally associated with folks living in poverty. That means that the children are generally underweight. The drinking water isn't properly purified. It could be unsafe bacteriologically. It could also contain other harmful substances, heavy metals, whatever. Poor sanitation and malnutrition. Poor sanitation, we know, goes along with infectious diseases like cholera and things like that, when we have waste that contaminates the fresh drinking water. Poverty makes the effect of chronic disease worse. So, for instance, pneumonia and diarrheal diseases generally don't kill children at such a high rate here in the United States or in developed countries, because poor children are already more vulnerable because their bodies are stressed because they're undernourished. So if you're already undernourished, when you get a infectious disease like pneumonia or a diarrheal disease, you're more likely to die from it because your body doesn't have the resources, the metabolic resources, to fight it off. Affluence changes the health risk factors for chronic disease. Better nutrition and sanitation mean that fewer children die of preventable diseases like pneumonia and diarrhea, and fewer actually even get them in the first place, just because nutrition and sanitation they're better in developed nations or affluent nations. In affluent nations, however, lifestyle diseases, and I put that in quotes, that's kind of a vogue term, right? Lifestyle diseases are a bigger threat. This means they're diseases that are caused by how you're living your life, right? So you've increased availability of tobacco in affluent countries, and tobacco smoke is a carcinogen. Folks in affluent countries tend to have a less active lifestyle. They use either public transportation or they drive their own personal vehicles more, which leads to a sedentary lifestyle and can lead to heart disease and obesity. Poor nutrition, and I don't mean undernutrition. We talked about the difference between malnutrition and undernutrition. In affluent countries, people tend to eat a skewed balance of nutrients. They're eating too much fat, too much meat, too much sugar, and they end up with malnutrition rather than undernutrition, and then just simple overeating, eating too many calories in a day, and gaining weight as the result of that. Heart disease, high blood pressure, diabetes, obesity are much, much, much bigger threats in affluent nations. I thought this was an interesting graph. This is high income versus low income countries and the percentage of the population that dies in these various age groups. If you think about your population pyramids that we did back in the first semester, these are the age groups that fit along with those population pyramid chunks, although some of them are larger than that. So 0 to 4 is generally considered early childhood, right? And then normally in a population pyramid, you'd have like 5-year segments. So you'd have 5 to 10. And you notice that this one actually goes for 15 years. So, and then this one right here, 15 to 59, that's a huge chunk, right? That would be much larger than a population pyramid would break it down. But these are roughly, the chunks that we're looking at here. So take a look at the difference between this. In high income countries, 56% of the population dies between 15 and 59 years. And there's various reasons for that. So you'd say that the majority of people are dying between 15 and 59. So take a look at infant and child mortality, which is this blue section right here. It's at 5%, right? And infants and children die for a variety of reasons. Infectious disease, they can die from genetic abnormalities. They can die from accidents. They can die from abuse, those kinds of things. And then we've got a 4% for 5 to 14 years. So this is people from 0 to 14 years. Total 9%, 5 plus 4. Okay, and then of course you would expect that the over 60 years would be a fairly large percentage, 35%, right? So think about when we're looking at survivorship curves, right? This would be a survivorship curve for high income countries where most of the children are surviving. And then as the lifespan goes on, we end up with a fall off of an increase in mortality towards the older years, right? Normal human survivorship curve. Take a look at low income countries, however. Things are a little bit different there. 0 to 4, 31%. Way, 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 way larger than both 0 to 4 and 5 to 14 combined for high income countries. So if you add 8% to that for the 15 to 14, the 5 to the 14 years, we've got like 39% of your population is dying before the age of 14 years. And again, if you think about developing nations, they have a very wide base in their population pyramids and a very small top. The reason that this is the case is that in order to keep up with replacement level fertility, they got to keep having children because the children die at an early age. So your greater than 60 years here is much, much smaller. And the reason why is because people don't often get to over 60 years. And then again, your 15 to 59 years is 48%. That is the bulk, but that's also a large span of years that they've included there. So anyone from the age of basically sexual maturity all the way up to 59 years. So that's a large chunk of the population. But it's that difference in mortality for children and youth up to the age of 13. That's so very striking there. And of course, in apes, you need to know the reasons why. So pathogens have evolved many ways of infecting humans. Transmission of pathogens can occur through the food you eat, the water you drink, the air you breathe. It can occur through other humans, especially contact in crowded places with other humans, and through contact with either domestic or wild animals, which we call zoonotic transmission. An epidemic occurs when a pathogen causes a rapid increase in disease in a regional or a national area. A pandemic occurs when a pathogen causes a rapid increase in disease over a multinational or on a global scale. Pan means across, so pandemics are going to be wider spread than epidemics. Some diseases associated with epi and pandemics include cholera, hepatitis, plague, malaria, and tuberculosis. Now, these are historical diseases associated with epi and pandemics. Some of them are still active in parts of the world, and some of them are not nearly as much so. I'm thinking particularly about the plague, but the rest of these remain pretty heightened threats, especially in developing nations. So we're going to talk about each, well, with the exception of cholera, we're going to talk, and I take that back. We're not going to talk about cholera or hepatitis individually. We're going to talk about plague, malaria, and tuberculosis, however. All right, so this is the plague. Yep, it's caused by the bacterium Yersinia pestis. That is a bacteria that is carried by fleas and spread by rats. So fleas are the main vector here. It actually can be spread by any mammal that's infected with the fleas that contain the bacteria, but it has historically been spread by rats. It is also known as the bubonic plague or the Black Death. Outbreaks of the bubonic plague or the Black Death wiped out large sections of the population in Europe over and over and over. There was plagues throughout Europe. Historically, lots and lots of instances of this. Modern antibiotics have pretty much wiped out the plague. There are still local outbreaks in India and in the American Southwest. However, plague is treatable. I mean, it's a bacterial disease. So that figure there, it's a wax figure, that is not a real human being. And they're showing the effects of the bubonic plague on one half of that wax figure. The other half is not showing those effects there. It was a pretty gruesome disease, and it caused a very, very painful death. Malaria, the scourge. Malaria is caused by protists of the genus Plasmodium. There are several different species of protist in that genus that causes malaria. Mosquitoes are the vector for human transmission. Malaria causes recurrent flu-like symptoms, and the mortality for malaria comes again from the fact that if you're already undernourished, because you live in a developing nation, you don't get enough to eat, then this protist can actually cause death, especially with the fever that comes along with malaria. Undernourished children die. They're too weak to fight the disease. Mosquito control programs, including the spraying of insecticides, which includes DDT, have been very effective at reducing malarial mortality. There's, mosquito eradication for many reasons. Mosquitoes, there's emergent diseases, Zika, West Nile, and those are also mosquito-borne. So the eradication of mosquitoes is a pretty big deal and can control a lot of both historic and emergent diseases. Tuberculosis. Tuberculosis is caused by the bacterium Mycobacterium tuberculosis, and those should both be italicized because it's a genus species name. Tuberculosis is spread by respiratory particles in the air, sneeze droplets, cough droplets, stuff like that. Tuberculosis can be cured by antibiotics, but here's the deal. The antibiotics have to be taken over the course of a year. It's a very long treatment of antibiotics to cure tuberculosis. So failure to take medication correctly has led to the development of several antibiotic-resistant strains of tuberculosis. And this is particularly true in Russia and Africa. They have a big problem with antibiotic TB. Most infections occur in the developing world due to a lack of medication or an improper use of medication. However, exposure to smoky indoor cooking fires. So if you remember when we were talking about indoor air pollution, in the developing world, a lot of people cook indoors in poorly ventilated areas using something that makes a smoky fire. The inhalation of those particulates and the smoke from those fires can weaken someone's lungs. And so when they're exposed to the tuberculosis bacterium, it more readily takes hold because the lungs are already damaged. Again, the World Health Organization estimates that so TB is a big problem in developing nations, and they estimate that there's a synergistic effect between the weakened lungs from indoor cooking fire along with this TB infection that's kind of driving the severity and the widespread scope of TB infections in the developing world. Okay, so we talked about some of the historic infectious diseases. Let's talk about some emergent infectious diseases. Emergent infectious diseases are those which are either new, meaning that they haven't been described before, or they haven't been common for at least the 20 years prior. Many emergent infectious diseases are what I call species jumpers. It's not just my term. It's not specifically used in the book, but you'll hear it. Species jumpers are pathogens that previously only infected animals or some other species, but have been able to mutate and they're now infecting humans. These include HIV/AIDS, Ebola, mad cow disease, bird flu, SARS, and the West Nile virus. So emergent disease transmission rates have increased in the last 20 years. Why? Well, it's easy to hop on a plane and travel anywhere in the world within 24 hours. So if you are infected with something and you don't know it yet, hop on a plane, travel anywhere within 24 hours, boom, you are able to infect a whole new population of people. Many people are living in close proximity to animals. Again, we call that zoonotic transmission. And this is particularly the case. Bird flu and swine flu were passed because people were living in close proximity to animals. Global climate change. So as the world warms, pathogens that were once restricted to tropical areas and vectors as well, mosquitoes particularly, are able to expand into areas that were once too cold or too dry. Let's just talk about the United States. In the United States, there was a freeze belt where the fleas died every winter north of the certain freeze belt. As the world warms, that freeze belt is getting further and further north. So we end up with flea infestations, fleas can pass disease. We end up with these flea infestations further and further north, or even northward. They last for longer and longer until the freeze occurs. And again, mosquitoes, remember, are killed off each year by cold weather. But if you don't have cold weather, or if your cold weather comes, later or doesn't last as long, then you've just got more chance to get a disease. There's also just been a increase in global population. More people living together equals increased rates of disease transmission. There's a lack of vaccinations, and this is kind of twofold. In developing nations, people don't have access to vaccinations. And in developed nations, there's been kind of hysteria about the false connection between vaccinations and autism. False. So people have not been vaccinating their children, and we're seeing outbreaks of infectious disease that were rare previously. Antibiotic resistance and rapid mutations in pathogens has led to emergent diseases gaining a foothold where they might not have previously. Let's talk about HIV. HIV causes a syndrome called AIDS. HIV is the human immunodeficiency virus. AIDS is acquired immune deficiency syndrome. It's a retrovirus, meaning it uses RNA rather than DNA. It's spread via sexual contact, blood transfusions, unclean intravenous drug needles, and it can be passed mother to child during childbirth if the mother is infected with HIV. HIV is a species jumper. There is a similar virus called SIV, which is simian immunodeficiency virus in chimpanzees, but it's not fatal to chimpanzees. It makes them ill, but it's not fatal. HIV can be controlled but not cured with antiviral drugs. However, antiviral drugs are expensive and they're often not available to those in developing nations. Ebola, first discovered near the Ebola River in the Democratic Republic of Congo, hence its name. It is a virus. It's viral transmission. Again, it's a species jumper. It jumped from chimpanzees, and Ebola causes high mortality in chimpanzees as well. Its probable origin is fruit bats, although that's still being investigated. Like, where is its species reservoir? Probably fruit bats, but we're not 100% sure. So Ebola is very, very, very dangerous. It kills up to 90% of its hosts within 2 weeks. At present, Ebola can only be controlled with experimental drugs. We do not have a surefire treatment for it, which is why when there's an outbreak or an epidemic of Ebola, people are quarantined. And it's highly infectious. Mad cow disease, also known as bovine spongiform encephalopathy, or BSE. Mad cow disease is an interesting one. It is caused not by a traditional disease vector, not by a traditional pathogen like bacteria, viruses, or a protist. It's caused by prions. Prions are misfolded proteins. They're proteins where their quaternary structure has become misfolded. And in that process, they actually are causing disease. They've become pathogenic. These prions can actually induce other nearby proteins to also misfold, and that's what causes the disease. Prions is what prions are and how they how they cause disease is a really kind of a cutting edge area of biology. It's fascinating if you're ever interested in it. Now, mad cow disease is similar to the scabies disease in sheep. It may be a species jumper. Again, this is still being investigated. So transmission in cows occurs because cows were fed ground up remains of infected cows. Yeah. Cows are herbivores, guys. And in the United Kingdom, in Britain, to add protein to the cows' diets, the farmers were grinding up dead cows and putting it into the feed they were feeding their cows. Well, the cows they ground up happened to have BSE, and they passed it to the herd through this infected dead cow food. The whole series, the whole string of that makes me crazy. Whoa, my gosh, gross. So it affects the central nervous system of animals. It leads to a loss of coordination and eventually tremors, loss of coordination, and eventually death. Humans who eat contaminated beef from animals who have BSE, particularly if they eat parts of the brain or the spinal cord of the animal. And people do eat cow brains and calf brains. Also, they eat the tail, like oxtail soup. Any of that stuff is going to be you're going to be at high risk of contaminating BSE if you're eating it from an infected cow. Humans develop a variant of BSE called Creutzfeldt-Jakob disease. That's the human version of BSE. 166 people in Britain have died of CJD, or Creutzfeldt-Jakob disease, since the mid-1980s. There's now a ban, of course, on feeding your cows, ground up cows who have BSE, but it's a bad thing. Okay, swine flu. Swine flu, H1N1. Again, species jumper. It normally only infects pigs. But it has jumped in the past. So the 1918 Spanish flu pandemic, and it was a pandemic, it was worldwide, it killed 100 million people across the world. That's a big number. It was an H1N1 variant, and it jumped at that time. And epidemiologists, people who study the transmission of disease, they believe that it's jumped multiple times in the past. The recent outbreak in 2009-2010, I had it at that time, killed over 18,000 people worldwide. And again, it was also a pandemic at that point because it was killing people worldwide. The bird flu is caused by the H5N1 virus, also a species jumper. It's a bird virus, and this one hadn't been known to jump species before. Humans have no evolutionary history with H5N1, so mortality was pretty high. It was about 50% with the bird flu. The only way that we could stop that transmission, anytime there was bird flu detected in a flock of birds, the entire flock had to be destroyed. They had to be killed. SARS. SARS stands for Severe Acute Respiratory Syndrome. It's also a bird disease, but it's not H5N1. SARS is a coronavirus, and coronaviruses also cause the common cold in humans. So humans are not unfamiliar with coronaviruses, just not this version of the coronavirus. Coronaviruses are highly contagious. So SARS has a high infection rate, but only about a 10%, I say only, mortality rate. So you can die of it, but the viral transmission is higher than the mortality rate. And this was first seen in Asia, parts of Asia. West Nile. West Nile lives in birds, that's its reservoir, but it's transmitted by mosquitoes. It's a mosquito-borne virus. It causes inflammation of the brain, leading to eventual death. Now, can you fight it off? Yes, you can. I actually had West Nile virus. I actually got it through mosquitoes several years ago. I was very ill, went to the doctor, and I recovered. I felt like I had a really bad flu. So clearly I didn't get inflammation of the brain. People do get it. People do survive it. People do get it. People die from it. So I think it just depends on, how bad your infection is and how strong your body is to begin with to fight it off. Increased mosquito control measures have reduced the threat of West Nile virus, and West Nile virus kills birds as well. So when people in communities start finding dead birds, they usually test the birds for West Nile virus to see if they are infected, and if they are, they begin mosquito eradication procedures. Now, one virus that's not on this slide, this PowerPoint, because it wasn't in your textbook, is the Zika virus. Zika virus is also a mosquito-borne virus. The Zika virus causes microcephaly. It's particularly dangerous to women of childbearing age because if they are pregnant during their Zika transmission, the baby can be born with mental retardation and brain defects. Microcephaly means a small head where the brain isn't developing correctly. The virus, and this is again ongoing research, the virus somehow induces the brain to not develop properly. And it's been a huge problem in tropical areas of Brazil. They've had a lot of Zika virus outbreaks that have caused subsequent outbreaks of microcephaly and brain damage in infants. Okay, toxicology and chemical risks. Chemicals can have many effects on humans. No, that's a big statement. Chemicals are currently grouped into neurotoxins, carcinogens, teratogens, allergens, and endocrine disruptors. And I know you probably know what a neurotoxin and a carcinogen and an allergen is, but we're going to talk about each one of these in turn. Okay, neurotoxins. Neurological, nervous, they are toxic to the nervous system of animals. Exposure to a neurotoxin leads to brain damage and other neurological effects. These can be insecticides. And the biggies that we talk about, of course, in apes are mercury and lead. Remember methylmercury poisoning in Minamata? That was a neurotoxin. Low level exposure can cause mental retardation in children. You can end up with tremors, all kinds of central nervous system problems. Carcinogens are chemicals that cause cancer. Carcinogens that directly damage the DNA are called mutagens. What kinds of things are those? Well, asbestos. Asbestos, tiny little fibers can get in your lungs, cause mesothelioma, which is a type of lung cancer. Rare, and of course caused by the asbestos fiber lodging down in your lungs. Arsenic is a carcinogen. Radon, formaldehyde, and tobacco smoke. Tobacco smoke, common carcinogen, but yep, it causes DNA damage in lung cells. Teratogens interfere with the normal development of embryos and fetuses. Thalidomide, there's actually a picture of a girl who was affected by thalidomide in your book. Alcohol is a teratogen, which is one of the reasons why they tell pregnant women not to drink when they are pregnant, because alcohol can disrupt the normal development of embryos and fetuses, and many other prescription and non-prescription drugs. You're really not supposed to ingest anything other than like food and water when you're pregnant. The little girl in this picture here, this is a little girl from Ireland. Thalidomide was prescribed to women in the 60s and 70s for morning sickness. Women tend to get nauseated when they're newly pregnant, and so thalidomide was prescribed as a morning sickness medication. Unfortunately, it also caused severe birth defects, particularly in limb development. So thalidomide babies had missing or malformed arms and missing or malformed legs. So there's a kind of a whole generation, particularly in the UK and in parts of the United States, where women were given thalidomide for morning sickness, and birth defects were the result. Allergens cause an allergic reaction in susceptible people, and allergens can actually lead to death. So bee venom is an allergen for some people. Milk, peanuts, so food allergies. So you can have insect allergies, you can have food allergies, and then you can have medication allergies like penicillin or codeine. People react badly to different substances, usually insect bites, foods, and medication. Endocrine disruptors. Now, we've talked about these before. Endocrine disruptors are chemicals that interfere with the normal functioning of the body's hormonal system, or a specific hormone. Endocrine disruptors bind to cells. They're usually mimics of a certain hormone, meaning that they can fit into receptor sites on cells and cause the cells to respond in non-beneficial ways. Hormones from birth control pills in particular end up in wastewater, which leads to hermaphrodism in fish and amphibians, meaning that their sex organs are indeterminate. You've got the development of ovaries in externally male fish, you've got the development of sperm in externally female fish, which causes massive problems with reproducing. So generally speaking, endocrine disruptors lead to reproductive harm. One that we've talked about a little bit as well is bisphenol A, or BPA. It's a chemical that's found in plastics, and it is also an endocrine disruptor. It's an estrogen mimic, so it mimics the effect of estrogen. So if you are consuming food, consuming liquid from plastic containers that contain BPA, then you're getting a chemical in your body that mimics the effect of estrogen. And too much estrogen in a male body can cause feminization and other undesirable changes. And too much estrogen in a female body can actually cause cancer, particularly breast cancer. Oh, by the way, the number one source of BPA is actually cash register receipts. Cash register tape is treated with BPA and to make it smooth. And that BPA it's a very thin layer. It can actually rub off, slough off, and get in your system from your fingers. So that's the biggest source of BPA contamination, because we've kind of phased it out of a lot of plastics, but it is still in cash register receipts. Scientists perform dose-response studies to assess the risk of chemical exposure. So if we're trying to figure out what's the risk to someone of a chemical, we perform these dose-response studies. A dose-response study exposes an organism to different amounts of a chemical and then observes a variety of possible responses, including mortality, which means death, and changes in behavior or reproduction. Sounds like fun, huh? Experiments that expose an organism to the chemical for a short period of time are called acute studies. Studies conducted for longer periods are called chronic studies. And this goes with acute disease and chronic disease. That's what those words mean. Short period, long period. This is an LD50 graph. We will be creating these. You have to know how to read these. On the bottom of this graph, you can see that there is the dose, and it's on a logarithmic scale. If you look closely at the scale, you'll notice that the units are not equal, right? They get closer together as you get further up in the dosages. And there's something called logarithmic graph paper that you guys will be using in order to create your LD50 graphs here. And on the y-axis here, you have your percent responding. So the LD50 is the dose at which 50% of that population has died. LD stands for lethal dose. So what you do is you find out where your 50% is, you drop it down to your x-axis, and you look at your dose. That's your LD50. We're going to be practicing with this in class. You'll do an LD50 lab. When the data from dose response studies are graphed, they generally follow an S-shaped curve, which is what you just saw in the previous slide. LD50 is lethal dose 50%. It's the dose of a toxin that kills 50% of the target individuals. The threshold dose is the dose at which adverse effects are first experienced by the population. So that's the what they call threshold or threshold of toxicity. I've heard it called both things, toxicity. Okay. That's the lowest dose at which you start to see adverse effects on an organism or the population of organisms. Sublethal effects occur when a toxin doesn't actually kill an organism, but causes effects on its behavior, physiology, or reproduction. So sublethal effects would be seen at a threshold dose. ED50, which is the effective dose 50%, is the dose at which 50% of the individuals experience harmful but sublethal effects. So toxic substances, they are regulated and controlled by the EPA under the Toxic Substances Control Act of 1976. However, now cosmetics are not under that at all. Cosmetics are a totally separate thing, and actually there's not a whole lot of legislation that talks about what can be in and not in those things. And pesticides are not covered under the Toxic Substances Control Act either. Pesticides are regulated under the Federal Insecticide, Fungicide, and Rodenticide Act of 1996. That's FIRFA. So yeah, it wasn't until 1996 that we actually regulated pesticides. Put that in your pipe and smoke it. My goodness, that was a long time of unregulated pesticide use. Toxic substances are usually tested on 4 organisms: a bird, a mammal, a fish, and an invertebrate. So the bird is usually a pigeon that they test on, a mammal, which is usually a mouse, a fish, which they usually use trout, and an invertebrate, which they usually use water fleas for that. Notice there that we're not testing on amphibians or reptiles. This is not a good thing, because what we do know about toxic substances is they often end up in the water. And so a lot of the substances, especially pesticides, disproportionately are harming amphibians and reptiles, but we don't have the data that tells us exactly how. We have to figure it out when there's some kind of problem with an amphibian or reptile population in the wild, and then go, oh, like what's doing this? So yeah, currently they only test on 4 different types of organisms: birds, mammals, fish, and invertebrate, which leaves that whole how does it affect amphibians and reptiles kind of up to chance, which is not good. So for most animals, safe exposure to a pesticide or to a toxin is calculated by taking the LD50, the lethal dose 50, and dividing by 10. And they say, well, okay, a 1/10 of this LD50 should be okay for chronic exposure, not to cause ill harm. Now that also depends on the threshold dose and the sublethal effects. So one of the things about LD50, and we talked a little bit about LD50 when we talked about our pesticide agriculture unit. The more potent a substance, right, the lower the LD50, meaning the less of it is needed to actually kill the population. The less potent a substance, the higher the LD50, meaning that there's more of it that's needed to kill 50% of the population. So again, with safe exposure, it depends on the threshold level as well. If the threshold level is very low, meaning that it only takes a little bit of the substance to see a negative or a sublethal effect, then safe exposure might not be LD divided by 10. It might be LD divided by 50 or some other much larger denominator there. Okay, retrospective versus prospective studies. Retrospective studies monitor people who've been exposed to a chemical in the past. The boy in the picture was exposed, well, actually his parents were exposed to methyl isocyanate gas in Bhopal, India. And it caused reproductive harm to them. And as a result, this child ended up with birth defects. So one of the biggest retrospective case studies has been the study of the accidental release of methyl isocyanate in Bhopal. The Bhopal gas disaster is one that you need to know as a case study for apes. It's well detailed in your book, so I'm not going to go into every detail of it. Union carbide, there's a union carbide pesticide plant in India, and there was an inadvertent release of a very, very toxic gas called methyl isocyanate. Immediately after the release, 2,000 people died, and about 15,000 in the weeks following. It caused blindness and respiratory distress immediately. You'll see people in your book sitting with their eyes bandaged. That is the result of the gas. So years later, even now, up to 100,000 people are still suffering various health effects, including genetic abnormalities, an increase in stillbirths, an increase in infant mortality, kidney disease, and learning disabilities due to the damage that this release of methyl isocyanate caused in the population. Terrible, terrible, terrible disaster. It happened in 1984, and people are still seeing the effects of it. Prospective studies, on the other hand, monitor people who may become exposed to a harmful substance in the future. So they're monitoring to see kind of, in a lot of cases, how lifestyle, what they are currently doing in terms of what they eat, how they live, might interact with a toxic substance in the future. And this leads us into synergistic reactions. Synergistic reactions are when one or more risks come together and they cause more harm than you'd expect them to harm based on their separate effects alone. And this is a concept borrowed from toxicity and toxicology studies. There's a graph of it here. So let's say you have substance A and substance B, and those are their toxicity levels taken separately. Well, if you add the area under those two curves together, you'd expect to get this dotted curve right here, right? A plus B equals C, yes? However, what we often find, especially with living bodies, is that when you combine two risk factors, they often cause an outsized risk, disproportionately large risk. And it depends on the organism that you're looking at as to why. But once a toxin has impaired an organism, then an additional toxin is going to impair the organism more greatly because the system's already weaker. So again, you can see that synergistic effects would have an outsized increase in risk effect or toxicity effect. The movement of a chemical through the environment is based on its solubility. The solubility is the measure of how well it can dissolve. And usually we're talking about dissolving in either water or in oil. So we're talking about water-soluble or fat-soluble chemicals. The greater the solubility in water, the greater potential for a chemical to run off, to leach into surface and into groundwater. The greater the solubility in oil or fats, the greater potential for a chemical to bioaccumulate and biomagnify, because fats are present in the tissues of animals. Bioaccumulation occurs when the concentration of a chemical increases in an organism over time. The rate of accumulation depends upon, number one, the amount of the chemical in the environment. The greater the amount of the chemical, the greater the amount of bioaccumulation. The uptake and excretion rate by the animal. If they're bringing in a lot into their body, but excreting it a lot, then it's going to be a lower bioaccumulation. Higher uptake, lower excretion, going to be higher bioaccumulation. Also, it depends on the amount of fat in the animal. If the animal has higher fat proportions, then they get higher amount of bioaccumulation. Biomagnification is an increase in the concentration of a chemical in animal tissues as that chemical moves up the food chain. Both bioaccumulation and biomagnification are tested concepts. So if you don't understand them, we can go over them in tutoring or whatever, but you got to know them. So the classic example of biomagnification is DDT. The insecticide was used heavily in the early and mid 20th century. It was banned in 1972. You have to know the book that started that movement, which is Silent Spring. Rachel Carson sounded the alarm about the dangers of DDT to bird populations, and as people began to investigate it, they realized that this was actually causing harm. So take a look at this graphic that's here. So DDT is a pesticide. Is it supposed to be in the water? No, but it can get in the water due to runoff. So let's say it's sprayed in an area for mosquito control. Well, it rains and the DDT runs off and it gets into surface water. And you can see that parts per million is really, really quite small. The DDT is then, it bioaccumulates in the bodies of the phytoplankton in the water and then up into the zooplankton. And because the zooplankton eat a lot of the phytoplankton, we end up with a higher percentage in the zooplankton. And as the zooplankton are consumed by small fish don't just eat one zooplankton, they eat multiple zooplankton. And so as the fish eat the zooplankton, it bioaccumulates and then biomagnifies in the tissues of the small fish. And as the small fish are eaten by the large fish, we now are up to 2 parts per million in the bodies of the large fish. Because large fish don't just eat one small fish, they eat multiple small fish. And here we go. Fish-eating birds. Fish-eating birds usually have the highest percentage of DDT in their bodies, percentages more than high enough to cause reproductive harm because, again, DDT, it bioaccumulates in the bodies of these organisms and then biomagnifies up the food chain. Fish-eating birds aren't just eating one fish, they're eating multiple fish. So they're getting the multiplier effect of the bioaccumulation in every single fish that they eat. So what does DDT cause? Well, it causes the eggshells of birds to thin to the point of breakage. If the eggshell is too thin, when the mother bird sits on it to warm them, the egg breaks. So this would be reproductive harm. The highest levels of DDT accumulation generally are found in fish-eating birds. And again, this is not nearly so much of a problem currently as it used to be because the pesticide in the United States was banned in 1972. It's still used in other countries, however. Persistence of a chemical is how long it stays in the environment. POPs, P-O-Ps. POPs are persistent organic pollutants. The more persistent a chemical is, the higher its chance to bioaccumulate, biomagnify, and cause toxic effects. DDT has a particularly long, quote, chemical half-life. It's still chemically active in the environment up to 30 years after its use. So, so it hangs around a very long time. It is definitely a POP, a persistent organic pollutant. Again, chemicals that stick around in the environment longer pose a greater threat to human and animal health than those that degrade quickly. Risk analysis. This is the final part of talking about human health risks and toxicology. So this is also in your book. It is leading causes of death. Now, the one in your book is for the United States. This is globally. And it's the size of the circle here is proportional to how many folks, in this particular case, for this graph, worldwide die of that effect, that risk factor, right? So by and large, the majority of people are dying of heart and circulatory disorders, followed by cancer, followed by respiratory disorders. That can be disease and just disorders. And you can see as that goes down, the relative risk from dying from certain risks, right? So as you go through here, right, nervous system disorders, digestive disorders, infections, non-transportation accidents, we are disproportionately afraid of things that we're statistically less likely to die of. Like, let's talk about murder. Murder. Look how small that circle is in comparison to heart and circulatory disorders, right? Do you live your life in fear of heart and circulatory disorders? Most people don't, right? But they do live their lives in fear of being murdered. And it's a legitimate fear, but that fear should be much smaller than the one where you say, oh, I'm going to exercise and eat healthy, because you're much more likely to die of heart and circulatory disorders than you are to be murdered. But we as a species don't do a great job of analyzing where our true risks, true mortality risks lie. And again, so what you just saw was an environmental hazard graph. It's anything in the environment that can potentially cause harm. Generally, humans suck at recognizing what things are actually harmful to them and what things are simply perceived threats. And I want to be clear, murder would be something that would be immediate and acute, right? If you're murdered, you're dead. However, like thinking of things like heart and circulatory disorders, those are things that are chronic, and and a lot of times they're lifestyle related. So, we say, oh man, I really should get out and exercise or whatever, and we don't do it, but we don't immediately think that that's going to have an immediately lethal effect, like, I don't know, being murdered. So we are really good as a species at recognizing short-term immediate threats, but not long-term effects and the accumulation of those risk factors to actual mortality rates. Okay, risk analysis. Risk analysis has three general phases. First, there's risk assessment. So you have to talk about the identification of the risk. What is the risk? The toxicity of the risk. Is it a high toxicity or a low toxicity? And the extent of the exposure. How long have I been exposed? How much exposure did I get? All those together is an assessment of the risk that you've been exposed to. We'll continue this on the next slide. The second part of risk analysis is risk acceptance. Am I willing to accept this risk or not willing to accept this risk? Right? You have to make a choice as to whether or not you're going to avoid that risk or accept that risk. And then if you accept the risk, then you're talking about risk management. How do I manage the toxic effects of the exposure to this environmental risk? Quantitative risk management is expressed by a very simple math problem. The risk is equal to the probability of being exposed to a hazard multiplied by the probability of being harmed if exposed. So if you have a high probability of being exposed and a high probability of being harmed, your risk is going to be pretty high. And again, if your probability of being exposed is high, but the probability of being harmed by that toxin or by that risk is low, then again, the risk is lower. Quantitative risk assessments bring together data from acute and chronic dose response data, retrospective studies, and prospective studies, which is why we talked about all those before we talked about risk assessment. Worldwide standards of risk are guided by two different philosophies: the innocent until proven guilty principle and the precautionary principle. I quote, innocent until proven guilty principle sounds really weird. So listen to this. So the innocent until proven guilty principle says the potential hazard should not be considered an actual hazard until scientific data demonstrate that it is. Okay? Sounds normal. There's all kinds of pitfalls to this. The precautionary principle is based on the belief that when a hazard is plausible but not necessarily certain, maybe we don't have complete data, we should take actions to remove the hazard. So let's talk about each one of these. Innocent until proven guilty. So you got a substance or a risk, and people say, oh, I don't know, that doesn't really seem safe, or, that might make you sick or something. And people say, well, data's out on that. We don't really have all the scientific data. So we're not going to regulate that hazard until it's been scientifically shown that the hazard actually is a real and actual hazard. This gentleman right here is following the innocent until proven guilty principle. Prove that it's not safe. The United States operates on the innocent until proven guilty principle. We will introduce substances into food, into water, into agriculture that have been untested. And we basically say, well, nobody's told us it is dangerous, and until we prove that it is, we're going to try it out. You can see where the logic goes in terms of, the fact that people are being exposed to stuff that may make them ill. Other areas of the world, particularly the European Union, operate under the precautionary principle, meaning that, well, we don't know. The hazard for this substance, it may definitely be there. It's plausible, but maybe not all the science is in on it. So perhaps we shouldn't use it or allow it into the public until it's further tested. And this woman right here is following the precautionary principle. It has to do, obviously, with regulatory mindset. It also has to do with lobbying by special interest groups. There's a lot of politics behind both of these, as you can imagine. But generally speaking, precautionary principle exposes people to far less environmental risk than innocent until proven guilty. The Stockholm Convention of 2001 created a list of 12 chemicals to be banned, phased out, or reduced. They called these the Dirty Dozen. The Dirty Dozen included DDT, which is already banned here in the United States, and PCBs, which are used in chemical manufacturing and plastic manufacturing, which they're regulated here in the U.S., but they haven't been banned or phased out. REACH was another European Union agreement, 2007, about the regulation of hazardous and potentially hazardous chemicals. Again, this right here, potentially hazardous, whoops, I'm moving stuff around instead of underlining it. Potentially hazardous, because the EU operates on the precautionary principle, they are looking to regulate substances that may cause harm. The United States does not operate on the precautionary principle. We have the innocent until proven guilty, so we'll throw it out there and see what happens, and we'll ban it after it causes effects, or if the science can show that it's dangerous. And that's the end.